Okay, so I'm going to kind of go through quickly what we went over in class as far as the identification part. So I'm going to skip through some slides. Anything I talked about in class is still fair game. I'm really going to just focus on the identification and then cover stuff that I wasn't able to get to in class. So let's see. Remember for the histology slide, you should be able to, if I have an image like this, you should be able to tell me that this is the testes would be the organ or the general. And if I ask, what is this entire unit called? You would say a seminiferous tubule. So if I had the pointer over here and I said, what is this entire structure called? You would say a seminiferous tubule. Now, if I have a pointer and it's along the periphery of a seminiferous tubule and I ask what's the specific structure, you would say spermatogonia. And remember, these spermatogonia are the stem cells which are capable of undergoing mitosis and meiosis. And as they undergo meiosis, they migrate through the seminiferous tubule. And when they reach the luminal surface and you see the flagella, this is our haploid spermatozoa. So spermatogonia around the periphery, these would all be diploid cells. And then they undergo meiosis, migrate towards the lumen, and when they arrive at the lumen and they have the flagella, they are now haploid sperm or spermatozoa, but they are incapable of fertilization. Remember the process of, so this is spermatogenesis, going from a diploid cell to a haploid cell. But spermiogenesis, which is the maturation of sperm, that takes place in the epididymis, or it finishes in the epididymis. And when the sperm leaves the epididymis, it is then capable of fertilization. At this point, it's just a haploid cell. So specific structure, spermatogonia, specific structure, spermatozoa. And then over here, these aggregations of cells that are in between the seminiferous tubules, those are interstitial cells of Leydig. And these are the cells that will produce male hormones, and the most prominent one being testosterone. Now, the Sertoli cells, these are these columnar cells that reach from the basement membrane or the periphery. So here's our testis. Then it's taking an individual cross-section through a seminiferous tubule, which are these highly coiled tubules within the testis. And then it's showing you up close what it would look like. So this would be the periphery, this would be the luminal surface. And the Sertoli cells are columnar cells that reach through the entire length of the seminiferous tubule, and these are supporting cells. And so what happens is, in the presence of FSH from the anterior pituitary, the Sertoli cells will mature and proliferate, and then LH from the anterior pituitary will go to the interstitial cells of Leydig and tell them to produce male sex hormones like testosterone. And then in the presence of FSH and LH, the Sertoli cells will stimulate the spermatogonia to undergo meiosis. Now remember, the spermatogonia will undergo mitosis first, and mitosis is going from one parent cell to two genetically identical daughter cells, right? And they're both diploid. So the reason why spermatogonia undergo mitosis is so that we can have a type A spermatogonia, which is left behind and it continues to be a stem cell. And then the type B spermatogonia, this would be a type B. And this, it will differentiate or specialize into a primary spermatocyte, still a diploid cell. And then this partially specialized primary spermatocyte would undergo meiosis 1. And at the result of meiosis 1 is that we have two secondary spermatocytes. And although it says it's haploid, it still has two sets of, now it have half the number of chromosomes, but they have identical copies of those, if that makes sense. If meiosis or mitosis doesn't make sense to you, if you've forgotten what that looks like, you should look up videos like on McGraw-Hill and look at the process of mitosis and meiosis if this confuses you. But meiosis has to undergo two divisions. And so these secondary spermatocytes will then undergo meiosis too. And as a result, we have 4 spermatids, all haploid, half the number of chromosomes. Then they'll begin to mature, and that's when we see a spermatozoa, or a sperm cell with the head, midpiece, and the flagella. But they are incapable of fertilization until they go to the epididymis. And so here's our testis. Now this white connective tissue, this is the tunica albuginea, and it invaginates down and forms these lobules within the testis. And then, there's hundreds of lobules, and then in the lobules there'll be the seminiferous tubules, which are very highly coiled, increase surface area, increase the amount of spermatogenesis that can take place. And so the spermatozoa, when they leave through the lumen of the seminiferous tubule, they'll travel to this rete testis, which is this aggregation where they all kind of accumulate. And from this rete testis, they'll travel up into the head of the epididymis, then through the body of the epididymis, and then through the tail of the epididymis. And what takes place in the epididymis is where the epididymis has that pseudostratified columnar stereociliated epithelium. That stereocilia is actually microvilli. And so excess fluids are reabsorbed. Sperm will be stored there for long periods of time until ready for ejaculation. And so it's a storage site for sperm. It also, as far as the secretions go, is not completely well known, but it's thought to aid in secretions that help with the maturation of the sperm or spermatozoa. And so when the sperm leave the epididymis, so when it's time for ejaculation and the sperm are leaving the epididymis, they are then capable of motility on their own. So before they arrive in the epididymis, they're incapable of motility on their own. After they leave the epididymis, they are capable of moving on their own, but sperm usually does not move on their own until they have entered into the female reproductive tract. So they rely on peristalsis through the vas deferens and the other channels so that they can save energy. So from the tail of the epididymis, when it starts to straighten out, this is now the vas deferens, and you can see the vas deferens coming up, entering this structure called the spermatic cord. And the spermatic cord descends the testicle within the scrotal sac. Remember, there are 2 testicles, and so the spermatic cord actually, suspends this testicle within the scrotal sac. Also contained in the spermatic cord will be the arteries supplying oxygenated blood to the testis and the epididymis, veins carrying blood, deoxygenated blood away, nerves to give a nerve supply, and then of course the vas deferens. So there's 2 spermatic cords, one coming from each testicle. Arteries, veins, nerves, and a vas deferens contained within it. Here's the epididymis, and I know that because we still see tubules, but if you look at the epithelium, this is clearly, there are multiple layers of cells in the seminiferous tubules. And although the nuclei of these cells are somewhat jumbled, you can tell that this is a single layer of cells. They're more just columnar cells, single layer of cells. And all of this stuff in the lumen, this would all be sperm. And so this would be, the specific would be pseudostratified columnar stereociliated epithelium. Specific would be sperm, and the general would be the epididymis. This is on high magnification, and you can really see those long stereocilia. And remember, stereocilia are actually not cilia. They're not for motility. They're very, very long microvilli. Remember when we saw microvilli in the small intestine, they're very short and stubby. This looks like cilia, but it functions more like microvilli as far as for reabsorption. Here's another image of also the epididymis, and so depending upon where you take your section from, the cells might look somewhat cuboidal. This is still, that pseudostratified columnar stereociliated epithelium of the epididymis. This is all sperm. So remember the mature sperm, we have the head, which contains the acrosome. The acrosome are hydrolytic enzymes that will help break down the outside layer of the oocyte for fertilization. And then we have our nucleus with half the number of chromosomes and a little bit of cytoplasm, not much at all. The midpiece contains lots and lots of mitochondria for energy production in a spiral fashion, and then the tail is a flagellum, which is capable of a whip-like or a lashing movement that will propel the sperm forward so that it can travel through the female reproductive tract. So semen production. Now, when it comes time for ejaculation, what happens is sperm will be released from the epididymis. It will travel through the vas deferens. And the vas deferens, if you see here, testis, epididymis, this is the vas deferens. It comes and it enters in through the inguinal canal. It courses over the pubic symphysis, or the pubic bone of the os coxae, loops around the bladder. This is the urinary bladder, loops around the bladder and connects on the posterior side to an ejaculatory duct, which a seminal vesicle also connects to. And you can see that here, vas deferens, up and over the bladder on the posterior side. And there's one on each side, so here's the other vas deferens. Here's a seminal vesicle, here's a seminal vesicle. The vas deferens will dump sperm into this ejaculatory duct, and the seminal vesicle will dump secretions which will aid in the production of semen, and the seminal vesicles will contribute prostaglandins and fructose. Fructose is a monosaccharide which can be used for the energy production, and then the prostaglandins will cause uterine smooth muscle contractions in the uterine wall to help propel the sperm to the fallopian tubes. So sperm from the vas deferens and then fructose and prostaglandins from the seminal vesicles will enter into this duct right here. There's one on each side. There's two. This is the ejaculatory duct, and the ejaculatory ducts merge into, or will dump their contents into, the urethra. And the urethra comes off the inferior side of the bladder. This chestnut-shaped gland that surrounds the first part of the urethra directly under the bladder, this is the prostate gland. Right here, this is the prostate gland. That would be the ejaculatory duct leading into the prostatic urethra. And the prostate gland dumps its secretions directly into the urethra, and it's a milky fluid which aids in the volume of semen, but also is going to, its main contribution is prostate-specific antigen, or PSA, and prostate-specific antigen actually will ensure that the semen does not coagulate after ejaculation, and so it continues to be liquid. A liquid is going to have move much faster than a coagulated substances. So PSA, or prostate-specific antigen, will liquefy the semen so that the sperm can actually travel better in the female reproductive tract. And it also aids in helping make sure that the semen is more alkaline. The vaginal canal is slightly acidic, and so if the sperm is alkaline, it will help neutralize the vaginal tract. Now this P-shaped